Okay, so this is my final project. I'm gonna go over, I guess, the processing and decision making for the bone marrow data set, and then the pancreas data set for the cell annotation tasks. So starting off with part one, which is the bone marrow, um, we have these two bone marrow data sets. To start out with, I did some basic filtering on read depth and number of unique features expressed. So here's the distribution for bone marrow data set one and data set two. And based on these plots, I removed cells with above 30,000 RNA counts, as these could be possible doublets, and cells with fewer than unique features, as these may be empty droplets or dead cells. And then, yeah, I'm just showing here that you should see a positive correlation between number of unique features and number of counts, which seems pretty standard here. I chose to use a couple different normalization settings using the log normalize. So I did 10 to the fourth. So log counts per 10K, log counts per 1 million, and then log counts per one. And I plotted the distribution of those here. And based on this, I decided to move forward with log count per 10K, as it seemed to preserve a good amount of variability in our distributions. And then I tried a couple of different settings for an iterative process for choosing the variable features. So I started off by just examining the top 2,000 most. And basically from this, I saw that it kind of seems like most of the features have below like 2.5 standardized variance. So ideally, I'd want to proceed forward with just the ones that are the most variable to reduce noise and preserve, I guess, variability in our data set. So I ended up choosing just the top 500 most variable features shown here for further analysis for both data sets. And then I plotted the principal components, 1 and 2, for each data set. So as you can see here, there's kind of, looks like 3 clusters maybe, 1 group of cells here, 1 along PC1, and then 1 in this corner. And similarly for bone marrow data set 2. And from the elbow plots, it kind of looked like the variability was starting to plateau around 15 PCs. So I proceeded forward with using 15 PCs for both data set to preserve the most variance in the data set. So next I tried the find neighbors, building the SNN graph. The author of the package suggested not to use K higher than the number of cells in the least populated cell type. So with some quick, I guess, napkin math, I assumed this would be 13. So I kept K as 10 and used 15 PCs. And then moving on, for the UMAP plots, I tried both Euclidean and cosine for distance metrics, and the results look pretty similar. The groupings for each of these colors tend to be in similar structures for both metrics. So I just, for simplicity, I went forward with cosine metric. And then I used MAST differential expression testing because they said that it handles single cell data better and that it models for dropout and sequencing depth. And basically, here are my marker genes. I used this cell marker 2.0 database for annotation as such, and then I annotated, and here's the UMAPs of my cell markers. So one thing noted here is CD4T, CD8T, and NKs tend to group together, and these cells are kind of known as being similar, so that's kind of additional validation that this clustering and annotation work somewhat correctly. So then for the pancreas data set, I pretty much followed the same iterative process. One thing to note here, there seems to be kind of a split between N feature and N count in how the cells group. The rest of the process is pretty much the same for pancreas, although at the end, when I went to annotate the cells, I found that many of the clusters had both sets of marker genes that I was looking for, which were these. So I was not able to confidently annotate the PP versus alpha cells. There's only one cluster for which it completely separated out based on my chosen marker genes. So for this one, it was a bit of a harder task. but the other one, it seemed like we were reasonably able to annotate cell types. So yeah, that's kind of my process and workflow in 5 minutes.
Thank you. Thank you very much. Okay, our next speaker is Azad. Please start sharing your screen
Okay. Get it ready.
and start your presentation. Yeah, I hope everybody can be prepared to share the slides because we are running short on time because we have a lot of presentations to do. So everybody has only 5 minutes. If you start late, then you will basically lose some time for your presentation. Yeah. Are you ready?
I am. Let me share the screen.
Okay. Okay. You
Uh,
have to stop by 3:40. Thank you. Yeah.
okay.
Yeah.
All right. So, basically for what I did, I'll just be showcasing the first bone marrow data set because just to put it in the slide, but I did the same thing for the second one and the pancreas data set. So, first I normalized the data and I used the scale factor of 1,000,000, and then afterwards I set the finding variable genes to 1,000, and I use a default process. And then afterwards, I scaled the data using default parameters. Um, I then ran PCAs with 50 PCCUs, and on the right is the plot that outputted the second bone marrow data set kind of looked the same. And then I ran an elbow plot to see what was the significant amount of PCs, but it was kind of hard to tell where I should stop or pick or which one was like diminishing returns. So I used a jackstraw to compute the significance of each PC. And for the first one, I stopped at 28 because of the afterwards the likelihood of the PCs being valuable just diminished. And so I also ran a gap statistic to see the optimal K cluster based on those PCs, and I got 12. And so I then ran the finding numbers with K equals 12 and with 28 PCs. And I did this with different values based on what the algorithms told me for the second bone marrow dataset and the pancreas dataset. And then I clustered and visualized the cells using UMAP, which is here, and I got 21 clusters from the algorithm. But then for marker genes, I ran find all marker genes on the dataset, and I then also created a database of potentially all known marker genes for each cell type, and then I went through and did a weighted score for each cluster's uh, marker gene, and whichever cell type came out on top with the weighted score was the assigned cell type I used, and then that is what I got here. And so from 21, we get down to, I believe, 13. And so yeah, that was my whole pipeline.
Okay. Thank you. I have just one comment to make. The gap statistics is to help you decide the number of clusters, but not the number of nearest neighbors in your KNN graph. Yeah, because they don't mean the same thing.
Okay.
Okay. Thank you. All right. So then let's move on to our next speaker. And Ryan, please start sharing your screen and start the presentation.
Hi, can everyone hear me and see my
Yes.
screen?
Yeah.
Okay, yeah, so I took a kind of a similar approach to Azad where I just want to share one data set because my pipeline was the same throughout all of my approaches. So here I'm just going to present on how I identify diverse cell types in the single cell bone marrow data. So initially I just put this brief motivation where why would it be interesting to study bone marrow data? So it's kind of interesting because this is a healthy patient. So it might be interesting to see how healthy bone marrow communities and functions might look. It's also helpful to observe lineage development, where it could be interesting to see how cells transition between active states or something like that. Then naturally, there's a lot of hematological diseases, so it could be interesting to see how uh, diseases might differ from healthy patients, and you can identify where those cell types might differ or how they're functioning differently. So overall, our goal is the question we want to answer is how can we identify the underlying cell types present in our data set, and we can leverage Seurat to do that. So I want to briefly go over the cell type discovery pipeline overall. And then I'll go over the parameters as I go through the steps in more detail. So of course we start with the raw data, which is just your gene by cell matrix with counts. And then I do a brief quality control, just like Azad and Atef. And then I ran SC transform to get my highly variable genes. So that will handle both normalization, scaling, and then the input is the variable features. And then from there, I run PCA, which will help inform my clustering decisions. And then further down, I also run clustering. And then I'm going to visualize with the UMAP, then identify marker genes. And then from the marker gene identification, I'll get the annotated cell types. So for the preliminary data processing, that's the quality control and SC transform. So quality control, what I chose to do was exclude genes that were present in fewer than 5 cells. So the reason why I filter out those genes is because it might make it harder. Those genes might be a little bit more noisy and unreliable, so it could be harder to identify marker genes from that set. Then I also excluded cells with fewer than 500 genes since I figured that could be indicating damaged or dying cells. So that reduced the amount of cells that I was working with by a small amount, and it reduced the amount of genes by a decent amount. But I thought that this parameters kind of balanced the amount of information I was gaining from each. So on the right, these are just the plots of the preliminary data, where I kind of filtered out the bottom left corner of these plots, or of the violin plot, I kind of filtered out the bottom, where those cells I kind of inferred that they were probably a bit low quality. So from there, I ran SC transform. And of course, I had these 16,000 total genes. And I decided to pick 2,000 highly variable genes. And I had tried higher values of 3,000 to 4,000, but I noticed that the amount of variance that seemed to be captured by the top genes kind of plateaued around 2,000. So I picked that value and kind of stuck with it. And then from there, I ran PCA. So I ran PCA, captured the first 50 PCs, and I plotted them in an elbow plot you see here. And based off of this elbow plot, I kind of decided that around 20 would be kind of an appropriate number to keep the dimensions, since it definitely flatlines out from there. Then from there, I ran clustering with both find neighbors and find clusters. I noticed that changing the K parameter value, I varied it from 10 to 50. And you get a lot of different clusters based off of what value you choose here, but I thought that 30 provided a good balance in combination with prune, SNN, and resolution, because I found that these 3 parameters in tandem will, vastly change the clusters that you get. And I ended up finding that based off of visuals, it seemed I was pretty satisfied with this set of parameters because the clusters seemed to be pretty well defined, and there seemed to be relatively low noise. For the algorithm, I chose all 3 different types of algorithms, but I noticed that they didn't change much, so I just stuck with one, which is just Louvain. And then from there, I visualized the created clusters with UMAP. These are just the parameters I chose because they looked good. And then from the results, I had 18 cell clusters out of the 21 possible. And then if you see on the UMAP, some of them are pretty well defined, like 14, 15, and 16 are pretty isolated, but others are kind of noisy and kind of combined, where you see 10, 8, and 6 kind of kind of blend together. So they could be similar cell types, or it could be UMAP being misleading, but it would take more downstream analysis to know. So that's why I went on and to identify marker genes. I did that with find all markers, and I decided to stick with the Wilcoxon test, even though I tested the student's T as well. So from there, I picked the top markers based off of their higher log fold change and percentage expression. Then, of course, they had to have a significant p-value. And then from I chose to annotate the cells based off of cell marker and PangLoud DB, where cell marker contains experimentally supported marker genes. That's this word cloud you see here for memory B cells. And PangLoud DB will take in a set of genes and it will output what it thinks that cell type is.
Yeah,
so I used
we should wrap up soon.
Sorry?
We should wrap up soon. It's already.
Yeah.
Okay.
Sorry.
Yeah.
So I kind of combined these, and here's just an example of identifying one cell type for hematopoietic stem cells. So pretty much identified these 2 marker genes present specifically in this cluster that was very strong. So this cell type was easy to identify. And then for other ones like macrophages, there are some genes kind of are expressed in multiple cell types, so it's harder to identify, but it could still be found. So overall, essentially, I just identified 18 cell types. There's some future things that could be improved, like maybe use more data or do more searches on cell type annotations to get higher confidence. But yeah, I'll keep working on it and kind of polish it up for all three of the data sets.
Thank you.
And thank you.
Yeah, that's great. Thank you. All right, so then Yihui, you're next.
Okay, let me share the screen. Okay. Okay, yeah. So here I'll just like briefly report the final project result I got. So for the part one dataset one, the parameter settings are like I mostly still use the defaults, but there are several things I think that is some parameter need to be adjusted specifically because of the task. The first thing is that the task gives like 20 give how to say like many cell types. So one thing I think is like including more highly variable genes in the analysis pipeline can like basically help to capture more key biological difference in rare cell types, because in those 21 cell types there are several cell types that are pretty relatively rare. And based on a similar reason, I also use really large resolution, like a resolution of 1, which is not like commonly used, but it helps to locate rare cell types because of same reasons about the more H3Gs I use. So I use like 3,000 features instead of 2,000 for fine variable features. In case that there are specific genes that can separate like specific rare cell types. Oh, sorry. And the other thing is that the dimensions, basically the number of PCs for finding the building the graphs are chosen based on the elbow plots. So it will differ. I would say it would be the only thing that differ between the dataset 1 and dataset 2, because I think dataset 1, dataset are coming from the same whole dataset. So I think it is more proper to keep the other parameters the same. Okay, so the annotation result of the dataset 1 is like here. So among them, myeloid progenitor and hematopoietic stem cell and plasma cells are, I would say, like uniquely identified in dataset 1 rather than dataset 2. And the next I will show the marker I chose. So you can see the classical monocyte cell has a unique expression of the CD14. And for CD8 T cell, the situation is a bit different because the markers of the CD8 T cells I chosen also has co-expression in other related cell types like regulatory T cells and natural killer cells, because they're like kind of biologically related to the CD8 T cell. So overall, I use overall three markers to combine to define this CD8 T cell cell type. Similarly, as the CD4 T cell is also like pretty complex cell type, I also use like more markers. There are unique markers like LEF1 and NEL2, but there are also like other markers that also has expressions in other cell types.
So Yihui, you have 1 minute.
Oh, okay. I will speed up. So regulatory T cell is CCR6. Natural killer cell is pretty unique. I will just go through this result. You can see I can identify those cell types by using multiple markers. Okay. And for dataset 2, the only thing changed is the dimension becomes 17 to 15. And the cell type that are unique is megakaryocyte cell. And the marker looks like this. And for part 2, because the alpha and say like PP cells are pretty rare in this data set, so I use an even larger resolution of 10. And it identified the alpha cells and the pancreas and the polypeptide PP cells. And it matched with the biological idea that the alpha cells and the gamma cells should be quite related. And the PPY expression looks like this. You can see it also had expressions in other cell types. And GCG similar to the alpha cells, and IRX like they are the markers I chosen. And after we got the annotation, we can find actually new markers of the alpha cells and PP cells. Yeah.
Okay, thank you, Yihui. We have to cut you off here, and we'll move
Sure.
on to our next speaker, Austin.
Yes, hello. Let's see, where is it? Okay, here we go. Okay, whoa. That's weird. Okay. Hi, so yeah, this is my final project, or what I've done so far, which in all honesty is not much. So yes, thankfully got I uploaded the 3 data sets. That's good. And so I've done the most work on the pancreas data set. So I'm very interested in looking at like the density of the RNA expression. So like I know that part of the pipeline is like normalizing the data and all that, but like I'm interested in like are there more cells where there's more like RNA counts on average per feature than others? Like can that help us? It was like that variable is not part of the data set, but I'm just wondering like if that's a form of normalization that could be useful. So in the pancreas data set, we see that there's like it's kind of skewed to the right in terms of the distribution.
What do you mean by density?
Right. So I'm just thinking like number of counts per feature on average. Just trying to see if that can tell. I understand that can be like a little misleading because like normally on a histogram density would be on the y-axis, but I just didn't want to like confuse by putting density on both axes, even because like they mean different things in this context. So and then okay, so this is like the pre-processing step, so log normalizing, finding the variable features, running PCA, clustering, etc. So based on the elbow plot, I noticed a very gradual decrease as you increase the PCs. So it looks to me like we can say up to 20 or at least like 16 PCs account for like a noticeable amount of variance, and then clustering it with UMAP. This is sort of the results. So we see a little cluster at the top, and so I'm wondering and towards the side. So I'm wondering if maybe those correspond with the sort of the outlier density values that we saw in the histogram. So I'm gonna like further investigate that. I think it's possible has nothing to do with it, but I just kind of want to investigate that for sure. And then for the bone marrow data, I got to looking at like the count per feature density. I should have probably clarified that in like my plots and stuff. So when I say density here, think counts per feature, I guess. So I looked at it for bone marrow 1 and bone marrow 2. So you'll notice for bone marrow 1 that in the upper far along the x-axis, there you'll see a little smudges of lines. So that means that this data is like skewed. So there are huge outliers in the bone marrow 1 data set as far as
You have 1 minute to finish.
okay, thank you. And then but for bone marrow 2, it doesn't look like there's like outliers. It looks like it's like pretty consistent. So yeah, that's all I got. Thank you.
Thank you, Austin. Okay, our next speaker is Yi Chen. Please start sharing your screen and start your presentation.
Hi everyone, my name is Yi Chen. This is my pipeline. I use a very similar pipeline for all 3 data sets, and I change a few parameters. So for the quality control, I'll use mean cells equals to 1 and mean features equals to 100, and use 10,000 for the scale factor. And then for the scale data, find variable features and PCA, I use the default parameters. And for find neighbors, I use the first 30 PCs for data 1 and data 2, and the first 20 PCs for data 3, because I think for data 3, the cell types, there are a few cell types, so the features are more similar compared to the first 2 data sets. And for fine clusters, I use 0.6 resolution for both data 1 and 2, and 0.1 for data 3. And for the next 2 functions, run UMAP and find all markers, I use the default parameters. And I also, for the cell type annotation, I use a website named Azimuth and use it as a reference for my cell type markers. And then I will briefly show a few results of my cell type annotation. This is for data 1. I find 16 clusters, and based on, because I have some knowledge about the immune cell types, so I use the marker genes I know, and I also use the azimuth as a reference to annotate the cell type. And this is the cell types for data 2. I have very similar cell types except for the megakaryocytes, which is not found from the first data set. And things becomes a little tricky for data 3. So first I find 6 clusters, and then you could see that cluster 1 are split into 2 parts. So then I use a function from Seurat called find subcluster. So I only perform that for the cluster 1. So then cluster 1 are subclustered into 2 clusters, 2 and 5. And then I use the feature plot. I plot the features of alpha cell and PP cell. And then I think cluster 2 should be alpha cells, and there is no PP cell that I found from this data set. And to confirm that PP cells are not found from this data set, I also print the top markers from class 4, which is here. And there are a lot of keratin genes and collagen genes. And then I use a website called Enricher, and to confirm that this cell type is not PP cell. So this is my final UMAP of data 3. So then this cluster should be alpha cell, and the rest are not annotated. So that's it, and thank you.
Thank you, Yi Chen. Yeah, very well-informed analysis. Okay, our next speaker is Jonathan. Please start sharing your screen.
Okay.
5 minutes. Yeah.
So I, like everyone else, I think I'm going to be sharing some similar information on sort of the standard Seurat pipeline. I'm drawing a lot here from the provided tutorial and using a lot of the default values provided. I was unable, just as an upfront, I was unable to install some of the libraries that Seurat uses. I think I'm having some issues with my C++ compiler, but this only seems to really impact the speed of the differentially expressed gene analysis and doesn't really impact the rest of my results. So during data input, I don't do any filtering for whether there's sparse genes or sparse cells, and instead sort of just using the default values there. I create these violin plots just to check whether there are cells here with strange numbers of features or counts. And it does seem from the scatter plot that there are some cells here that have reasonable numbers of features provided, but then have a very large number of counts. So I just do some filtering for cells that have fewer than 25,000 counts in the, or this should be feature or counts in the data. I perform log1p CP10K normalization. I wanted to look at other normalization methods, and I think I'm going to experiment with that a little bit for the second data set and the pancreas data set as well. But here, I think this looks fairly good. Moving forward with looking for highly variable features, I look for 2000. I think I might be able to use a smaller number here based on this plot, though I think sticking with 2000 gives me reasonable results later. I do then scale my data, perform PCA, just to visualize some of the results, because I think these plots look interesting. I just wanted to look at the loadings for some of the genes for a couple of the PCs, as well as plot some of the scores across the first 4 PCs. Performing dimensionality reduction after that, I opt to move forward with selecting the first 20 principal components for analysis. And I validate this also with a heat map where I look at the gene loadings per PC ranked by how much it impacts, how much the cell matters for that PC, or how much the PC matters for that cell, and validate that this is kind of hard to see in the HTML, but I am able to validate that farther than 20 PCs, there isn't that much structure in these figures. I perform cell clustering with, again, a lot of the default parameters. I use the Louvain algorithm and identify 14 clusters. I visualize my results using UMAP. I played around a little bit with some of these parameters to give me more or fewer visualized clusters here. I find that other parameter settings here either give me sort of like one big cluster or a very long, sort of like spindly ones. And so I find that actually the default parameters here give me results that seem fairly reasonable in terms of separating out different clusters, but then also become interpretable once I add in the cell type annotations.
So you have one minute. Yeah.
I find all markers using sort of the default parameters. This takes a long time, so I save my results. And then I just pull in marker labels and then one by one go through and using violin plots, manually annotate my cell labels. And I will sort of repeat this pipeline again for the rest of the data sets.
Thank you.
Thank you.
Thank you. Okay, our next speaker is Richard.
All right. Oh, can you see to my slides?
Yes, please start.
Okay, cool.
Thank you.
So I'll just briefly go over both the bone marrow and the pancreas cell annotations. So for part 1, we're looking at cell types in the bone marrow data set. And I think an interesting starting point is that all these cells are from kind of like the same lineage, starting from these progenitor and from these like stem cell. So that kind of informs some of my decisions and how I went about approaching the single cell analysis. Here I first compared dataset 1, dataset 2. Interestingly, I saw in dataset 1 there were some outliers in terms of the number of the RNA counts. So because this dataset is messier and actually because my results are worse, I'm going to actually focus on dataset 1 because I think it's more interesting. In terms of pipeline, I mostly kept the same workflow for both datasets, except the one thing I wanted to experiment was 2 different normalization methods. So from top to bottom, I used 5 cells and 500 features because I wanted to be more stringent in my cutoffs of like cells and avoid dropouts. And this is one I tried log CPM, which I might change back to 10 to the 4, and then scaling and then finding variable features versus SC transform in dataset 2. And I do the dimensional reduction clustering with more stringent pruning and then Louvain multi-level refinement because I think our cell types are highly similar, so I'm hoping to have like tighter clusters and better differentiation. And then I use find all markers to help with the annotation. Yeah, so the highest variance genes all seem to be functionally relevant to the lymphoid and myeloid lineages, so that just tells us that our cell types are probably, some of our cell types should be there. Not that interesting. For the selecting the number of dimensions, like PC variables, it was kind of hard to tell the elbow point, but I went with the less conservative threshold. This I'll just skip for time's sake. I found that the clustering method actually didn't matter too much, and I pretty much always got these same clusters. So now I'm just gonna go through some of my labelings. The bottom line is I think I still need to further refine my list of cell marker genes because I'm not super familiar with these cell types. But generally, I still think I get some distinguishment between these lymphocyte cells. CD4 identities could be within here versus CD8 over here, although they could also be part of these other cell types that are very similar. B cells, mostly 6, but I think I still have some problems where this identity, either due to the marker or clustering problems, it could be present in 11. And then the other cell types of plasma dendritic and then monocyte macrophage, and then these progenitor cells seem to be pretty well like locally close to each other or locally continuous. Oh yeah, sorry, we can go back. So this is just showing that like the general cell types are there, but I think especially for lymphocytes, I need to do some further refinements in my subclusters. And then looking at PC variables, it does seem that on PC1 and PC2, there is some differentiation of these different cell types. I'll just quickly go over the pancreas data set. Yeah, so we're looking between, these gamma cells and these alpha cells. The data for this one, I changed my workflow a little bit because now we're looking at 2 cell types only. So I still keep a stringent threshold. This is my normalization. I think the most interesting things are I set my clustering back to mostly the default method because I think there's less importance on like having tight clusters in this case. And then for now, I'm trying find all markers to distinguish the 2 cell types. I think my most important result is I think my data set has both of the cell types. Just looking at the simple PCA, it seems like 6 and 7. Oh, and just based on the marker genes, it seems like 6 and 7 here are the P the gamma cells, PP cells, and then these cluster 0, 2, 3, 4 as labeled by the clustering have the marker genes for alpha. However, clusters 1, 5, and 8 actually are not assigned. So
So
either
Richard,
I'm going.
we have to cut off your
Oh,
5
okay, sure.
minutes. Yeah.
Yeah, that's my ending as well.
Thank you. Okay, thank you. All right, our next speaker is Fei Yang.
Yes.
You're sharing your screen.
Yeah, I'm going to share it. Okay, can you see the screen now?
We're only seeing part of your browser, the header of your browser, not a full screen.
Oh, okay, let me. Sorry. Okay. Okay.
Okay, now we can see it.
Okay. So today I will introduce my pipeline for my final project, and I will use the part 1 as an example because the part 1 and part 2 is quite similar. So first is the quality control. I will filter out features that present in fewer than 10 cells and remove cells that express fewer than 200 genes. And here is a volume plot for the quality control matrix. And the first plot shows the number of genes detected per cells, and the second plot shows the number of RNAs captures, and then the third is the percentage of the mitochondrial gene expression. This is close to 0, so which is good. And then I also look at a scatter plot showing the relationship between the total RNA counts and and the number of detected genes for the data. And here is a strong positive relationship observed in both data sets, which saying that as the total RNA counts increase, the number of unique genes detected also increase. So which is expected in the analysis. And then to prepare our data, I first perform the log normalization and the scales for the data, and also identify the top 2,000 highly variable genes which capture the key biological signal in our data. And then I also perform another normalization called SC transform, which contains the scaling and highly variable gene selection automatically. And I will calculate the correlation between the gene variance and the gene means for both methods. And we can compare which process data we want to use for the downstream analysis. So from this plot, we can see that the SC transform have the ensure the variance is independent to the gene mean. So meaning that the gene with different expression level will have comparable variance, which is important for the dimension reduction and clustering. So I conduct a PCA from 1 to 100 to see how many PC we should pick. So for both data sets, I pick 25. And here is a plot showing the top contributing genes for the first two PC. After performing PCA, I also performing clustering. To process, the process starts with find the neighbors and then which identify the relationship between cells in lower dimension space, and I also use the find cluster to assign cells into distinct groups and use two. Okay, so
yeah.
Then I will just directly show the UMAP result and the thesis result. So here we can see that all the result has very well spread clustering, but I will use the UMass for the later because it maintains both the local and global structure. And here is the gene marker that I obtained from the online resources. And here is the final, I can directly show my final result. So I'm able to find out there's so many gene cell types, and some of these cluster is still unassigned. Okay, yeah, I think that's all. Thank you.
Thank you. All right, our next speaker is Aditya. Please start sharing your slides and start speaking. Hi, Aditya. Do we have you here?
Yep, I'm here.
Okay.
Present, share my screen. Okay, there we go. So here's my presentation on cell type annotation with Seurat. And so I'm just restricting it to the bone marrow data sets here for the sake of time. And I feel these are more complex and capture the essence of what I'm doing. And so there's like a pipeline that I'm using, which is based on Seurat. So you could use log normalization, and then there's tool single R, which does cell type annotation based on the log normalization and scaled expression values, and presents results. And these it uses like probabilities based on how well each row and their expression profile matches to a reference database. And so this will be used for the annotation. And this is sort of automatic. So I'll be doing a validation using the traditional pipeline where I identify highly variable genes, use PCA, and then Leiden clustering. And then a bunch of these I'll use other methods for visualizing these steps. So let's just start with log normalization. This is kind of what it looks like, very standard, just normalize these values and scale them. And then you can do the cell type annotation. And here you have them projected onto a UMAP. And you have around 16 types, and they're labeled on their location. And as you can see, they seem to capture a lot of the over the independent clustering. So you can see here, the B cells here, there's a region here that's independent and like not, we're separated, I meant to say, from other regions, and it's captured pretty well by this annotation. And then you can see the distribution of frequencies of types of cells or counts of types of cells. And so you can see T cells and NK cells and B cells, which are common in bone marrow. And so we see there are about 6 or 7 different cell types that are prevalent. And there are a lot of other types that are identified, but they were relatively small, like, less than 10. So we know that there are around 7 or so that are prevalent. The rest might be due to noise or some artifact. And that's where the validation comes in. And so first we take, after the normalization and scaling, look at the top 1,000 I chose highly variable genes or features. And I labeled the top 10 here. And with that, you can do PCA. And as you can see, there is some clustering seen in the PCA for the scale data set for the first bone marrow data set here. And you can also look at the variance contributed by each PC. And here there seems to be around 10. It levels off with the elbows. So for downstream analysis, I'll use that. And you can also look at these highly variable genes, how much each contributes to the PC, and you can see how much separation there is. And so 7 PCs seems pretty reasonable based on how much like each expression is distinct for each cell. And so then you can use Leiden clustering. So I used 0.25 resolution, which is kind of an arbitrary parameter, but it seemed to capture these independent these isolated clusters well without having divisions within them. And so there's around 7 clusters with a 0.25 resolution, which is what single R produces. And this lines up pretty well with the plot that you see here. So we can also look at the expression profiles for 6 of the top 1,000 highly expressed genes. And you can see they do seem to be distinct. For example, cluster 4 has which was cluster 4, which is
have to wrap up within 1 minute.
Okay, yeah, so cluster 4 is highly expressed. And then I also applied this to the second bone marrow data set, and got the following results here, which are similar in terms of their composition. So this kind of also shows like that it's as a second validation to show consistency between samples.
Okay, thank you. Thank you. Our next speaker is Allison. Please start sharing your screen and start sharing your screen and start speaking.
Okay, hi everyone. Today I'll be talking about the stats final project that we all did on the single cell data set. And so I'll be talking about an example workflow that I did for one of the bone marrow data sets. The first thing I did was look at quality control, and after looking at a couple different choices for these parameters, I decided on minimum cells 3 and min.features 200. And from there, I used SC transform to perform the normalization, scaling, and highly variable gene selection. And I used the default, which was 3,000 for the number of variable features chosen. After that, I used PCA to run dimensionality reduction, and I was following the SC transform tutorial, so I also just used the default for NPCs as they recommended. And then from there, to perform the clustering, I first did find neighbors, and SE Transform recommends that you can use a higher number of dimensions, so I went with 30 dimensions there. And for fine clusters, I ended up with the resolution of 0.5, which seemed to get a good amount of clusters that seemed reasonable. And then run UMAP, I also did 30 dimensions as well to stick with the 30 consistency in the fine neighbors. And then finally, to identify marker genes, I used find all markers, and I used the default test here as well. And for example, quality control, I checked the number of end-count RNA and end-feature RNA. And for the end-count RNA, the minimum was 524, which seems pretty good. And for the end-feature RNA, the minimum was 255. So most of these cells do seem to have a good amount of counts and features. And this is what one of the UMAPs looks like for the bone marrow 1 data set, where you can see some clusters are larger, like 0 and 1. Some clusters are smaller, but they're pretty distinct, like 12 and 13, which could indicate some smaller groups of cell types. And this is an example of one of the cell types that I identified, plasmacytoid dendritic cell. And I used cell marker 2.0 to get these marker genes listed here. And then I converted them to ensemble IDs. And this is plotting the expression for each cluster. And we can see that cluster 8 has pretty high expression of all the marker genes. And then this is on the right is the same data, but in the UMAP space. So we can see this little small cluster right here, which I've circled in red, is cluster 8. And over all the different marker genes has pretty consistently high expression compared to the other clusters. And so from there, I repeated that with all the other different cell types. And then from cell marker 2, and this is my final annotation where we see the T cells and natural killer cells are close together. And then also different types of B cells are next to each other. And then here was cluster 8. And that's my presentation. Thank you.
Thank you. Thank you. Okay, so our next speaker is Elise. Please start sharing your screen.
Can you hear me?
Yeah.
Okay. Hi everyone, I'm Elise, and today I'll be presenting the results I got for my cell type annotation project using bone marrow dataset 1. So I kind of wanted to just give a brief overview of the pipeline I use for cell type annotation. Just to save time, I'm just going to go through the steps like for the annotation after clustering. So basically, yeah, I ran PCA and UMAP clustering as well as marker gene identification using the pipeline that was presented during lecture. And then I basically installed and loaded the biomart package in R, specifically using the human gene database. And then I kind of basically went through each cluster in the UMAP object for bone marrow dataset 1 and accessed all the ensemble IDs of the differentially expressed or marker genes that were shown. And then using the biomart package, I was able to generate a table of these ensemble IDs as well as their corresponding gene names or gene symbols. And then afterward, for each cluster, I went through all of the genes and chose the most significant differentially expressed genes. And then I basically used these like top differentially expressed genes as input to a human gene database. The specific databases I used in this project were PangLaoDB and the Human Protein Atlas, but I mostly used PangLaoDB only because it helped me to like annotate more like multiple cell types. And so, yeah, so I basically would input these genes as input into the database, and then the output would be like a distribution of the different cell types that were known to be associated with these genes based on like multiple like published papers. And then from looking at the distributions, I was able to find what possible cell types could be, like whether there was one cell type or multiple possible cell types in each cluster. And then I basically went through each cell in the bone marrow dataset 1, and based on the cluster that it was assigned to, I was able to label each cell as that one cell type or one of the possible cell types using a sampling algorithm. And then I finally stored all these cell type labels as a vector in the metadata for the bone marrow dataset 1 Seurat object as per the instructions in the project. And so I kind of wanted to show more visually some of the results I got for bone marrow dataset 1. For the clusters, as you can see, this is the UMAP plot of all the clusters I got. There are 16 clusters numbered 0 to 15. And as you can see here, some of the clusters are like sort of more separated into like little like islands, I guess. But some of the other clusters are more like clumped together. I did try to go through and optimize or fine-tune hyperparameters of the models to like make the clusters as separated as possible while retaining cluster identity, like not having split clusters or like a bunch of clusters clumped together. But so it doesn't look perfect right now, but this is kind of the best I could get it to look in terms of like separation between clusters and cohesion of data points within clusters. And so I kind of just also wanted to show like a general distribution of the cell types that I got for bone marrow dataset 1. As you can see in this bar plot, the most common cell type was dendritic cell. The second most common one was a T cell, specifically CD8 T cell. And then it was like CD4 T cell, erythroid progenitor, and CD16 natural killer cell. The other cell types were less frequent, but nevertheless still able to be identified in the data set. So in total, I was able to identify 14 cell types, which was about 70% of the 21 cell types that were initially given. And so, yeah, these are kind of just the general results I got for the cell type annotation project. Obviously, there's a lot more optimizing or fine-tuning of parameters that could be done to make the results even better. But this is kind of like the initial results I got using the algorithm that I had in my project for my cell type annotation with bone marrow dataset 1. And yeah, that's it. Thank you.
Thank you, Elise. Our next speaker is Ornella.
That Joyce, yes. Yeah. Yeah.
Could you start sharing your screen? Yeah.
Sure. Sorry, can you see it? Yeah, sorry about that, but this presentation is going to be a lot shorter than everybody else. So I've been having some issues running Seurat. So I actually, for this presentation, I actually ran the Python version, but I'll make sure for the final presentation or the final project submission, I will run Seurat once I figure out what's going on there. But basically, since scanp is like the Python version of Seurat, one can say, I just try to follow the same pipeline as a lot of people have shared so far. Some of the pre-filtering that I did with the cell and the genes was like saying that I want the matrix to have at least 200 genes per cell and at least 3 cells as a second feature for the genes. As you can see for this first part, for the features, once you do this pre-filtering, the numbers change or like decreases, which will definitely affect the results. So I think that's something I'm going to keep also in the Seurat pipeline that I run this week. So I also did the same thing as other people, of course, normalization using default parameters, log transforming the data, as everyone else did. Also did some dimensionality reduction using PCA 50 components, and then some neighboring where the PCs were like 40. But I think here, everything I'm doing is very basic and very like random. So I think seeing what other people have done, maybe I should probably like find a better way of picking these parameters because yeah, all of this is random. And then the resolution using the latent clustering was 0.5. I saw someone else did 0.25. I don't know how that changes the results, but that was nice to see. And then UMAP for the final visualization. So for the first data set, if you just run the same thing, you kind of I already see like 15 clusters. The next thing I'm going to do is actually like labeling all of them, which that will be what will be my final project submission. Looking at the differential expressed genes, basically this is comparing like each cluster across all the other clusters, and you can identify some of these genes, but I haven't changed the naming properly yet. So that's also something that I need to do. Looking at the next data set, it's actually interesting that I'm losing like one cluster here compared to the other one. So I need to maybe investigate a bit more what's going on there. And the last data set also had like, I guess, less clusters compared to the other two. So I think it would be good to also look into like what exactly is going on there, which I haven't done yet. But real quick, Jessica, a quick question. I know the class focuses more on like running stuff in R. Have you looked into like running actual scan Pi and like what's the difference between them and like what do you recommend in general?
I think it's an open question. Yeah, some people use, I think the few use both, depending on whether the users are more familiar with R or Scam or Python. Yeah, I think it's an open question. Both are popular software packages.
Okay, yeah, because I'm used to Python. That's why I was okay. Let me just run Python and then get some result for this presentation, and then I will do what we're asked to do for the final submission. But yeah, this is it for me for now. Thank you.
Thank you. Thank you. Yeah, that's also good to know. Okay, so the next person is Nabil, the next presenter. Please start sharing your screen.
Okay. Can everyone see my screen?
Yes, we can see your screen well. Even
Okay.
if it's a black box, yeah, but now it's no longer blocking the view. Yeah.
Sorry, that's the Zoom thing. Anyways, yeah, I'm going to talk about what I kind of did. I follow a very similar analysis pipeline to everybody else, and I'll go through the bone marrow one data set. So first things first, I created the Seurat object, and then I wanted to visualize some QC metrics. So the ones I chose were the end feature RNA and count RNA, percent MT, which is the mitochondrial genes, and then percent ribo for ribosomal RNA. I looked at these plots. It's good to see there's no mitochondrial or ribosomal RNA. And then based on both of these violin plots, I kind of chose to subset my data based on the end feature RNA. I chose to go between 200 and 3,500. And then based on end count RNA, I went between 500 and 30,000. I then proceeded with log normalization, and I found the variable features, and I plotted these in this type of plot, just to pay attention, just identified genes that I should pay attention to in the more downstream processes. I then scaled the data, ran PCA, and I plotted heat maps of all the different PCs. I didn't find this particularly useful, but I found the elbow plot to be a lot more useful, and identified that I wanted to use the first 15 PCs for my further downstream analyses. So then I found the neighbors, and I did clustering as well to plot my initial UMAP, and I found there's kind of 4 rough main groups of cells. And then from here, I wanted to actually identify what types of cells each of these were. So what I ended up doing is pulling out the top 10 marker genes from all of these, and then looking it up in cell marker 2. And then I just manually kind of mapped the top marker genes for a specific cluster and found the names. So in the end, I think I found 8 different total cell types in the bone marrow 1 data set, and here's what my end UMAP looks like. Bone marrow 2 is kind of more of the same, so I'm going to skip to part 2, where the initial pre-processing was the same thing, but the task was slightly different in that we were given 2 different cell types and asked to figure out if these cell types existed or if we were able to find additional marker genes. So again, I did the same type of QC where I filtered on the end feature RNA and the end count RNA. I did log normalization, finding variable features, selecting number of PCs. Here, I think I chose all 20 PCs just because of what the elbow plot looks like. I did the UMAP, and things are looking a lot more scattered. And the main difference here was I actually used the PanGLAU-DB annotated marker genes. So essentially, all I did was download the data set and then filter to the specific cell types. I changed the names from the standard kind of gene naming convention to the ensemble one. And then I just filtered for the top 10 for each specific cell type. And within the provided data set, I searched to see were any of these found. And here's an example of a plot that I did for one of the 2. And I basically went and handpicked these genes to then annotate my end cells. And I found some counts, some instances of both alpha and the PP cells. I tried to look for more marker genes, but I was not able to find anything else. As we can see, pretty much everything else that lines up with no marker genes for both types of cells, I was not able to find in my data.
Thank you.
Yeah, that's all. Thank you.
All right, our next speaker is Rithvik. Please start sharing your slides.
All right, everyone. My name is Rithwik, and this is my final presentation. So the goal for this project was to go from single cell gene expression to cell labels. So a brief overview of what I did. I used SC Customize to get a bunch of QC metrics, including mitochondrial reads. I normalized with SC Transform, and I used this mathematical approach to determine the number of PCs. And then everything else is kind of similar to what everyone else did, and I'll go through and hopefully show some visualizations so that you can see the work that I was able to do for this project. The first thing that I found kind of interesting was when I initially tried to determine the number of mitochondrial reads, I got 0, and that's because I had ensemble IDs and all the mitochondrial read QC checks for MT as the prefix, so I had to convert first, and you can see how the QC that I chose was slightly different between bone marrow 1 and bone marrow 2, but I wanted basically a low percentage of mitochondrial reads in both. For the pancreas as well, there are actually a lot more features, so I actually changed my min and max features for my cutoff, but I kept the mitochondrial percentage the same. This is another like cool visualization that SC Customize allows you to do, where you can look at the percent mitochondrial and gene counts per each cell, and you can adjust your cutoffs. I didn't really use this to inform anything, but I just thought it was a neat visualization. So moving on to my PC analysis, this is what my elbow plots look like for bone marrow 1 and bone marrow 2 data sets. And instead of just like looking at it and trying to guess what elbow or where the elbow was, I instead did this thing where I looked at the percent change in variation between the consecutive PCs was lower than the threshold, and I used that to determine which PC to select. You can see I actually ended up with quite a few clusters, and at first I was a bit worried because of this, but then I realized that some of the cell types are pretty rare, like a previous presenter mentioned, so maybe this is actually a good thing. And this was at the end of an iterative process of me choosing a lot of parameters. When it came to the pancreas, I also had more clusters than there were cell types, but I think that's okay because this is still exploratory analysis. We haven't come in with a hypothesis that every single cell type is present in our data. Moving on to how I did marker genes. I looked at an online atlas like many of the previous presenters. You can see, for example, I used SDC1 for plasma cells, and that has a pretty strong signature for cluster 17. GNLY has a pretty strong signature for this cluster over here, and this gene has a pretty strong signature over here, and it's tied to PDC or plasma dendritic cells. And you can see this is my clustering for bone marrow 1. And diving more into that clustering for bone marrow 1, this is looking at those 3 genes I previously showed and where they show up. You can see, for example, that CLEC4C is related to the PDC cell. It's a marker, and you can see that its cluster it's highly expressed in that cluster. The same goes for the plasma cell and SDC1. This is just a violin plot instead of this expression dimension plot. And then you have the natural killer mass over here, which kind of matches with its marker, one of its marker genes. Moving on to bone marrow 2, you can see again how the cell clustering and cell type look like. My results are a bit more noisy here, especially in this area. Like some previous presenters mentioned, many cell types here are pretty related, and so it's kind of tough to separate them. And then for the pancreas, I was able to identify the alpha cell and PP cells, although there are a lot of other cells, basically cells that I wasn't really confident about. And even between these cells, I wasn't super confident, but there was enough variability in gene expression for me to make that decision. Overall, I did have a lot of clusters that I left unlabeled, and I think that that's okay because again, we didn't come in with the hypothesis that all of these cell types are in every single one of these cells, and you could have other cell types present. And so I think this is a really good learning experience working with different kinds of data and different types of cells, some of which I had not heard of before. Thank you.
Thank you very much. Yeah, it's a very nice summary. And our next speaker is Xingyue. Please start sharing your slides.
Can you see my slides?
Yes, we can.
Okay. So for the final presentation, for my project, I mainly have two different data sets. The first is from the bone marrow and the other is for the pancreas. And for bone marrow, I have two data sets, and I will start with a really brief introduction or background for the bone marrow. Bone marrow is the body's blood forming system responsible for producing red blood cells, white blood cells, and platelets. And for the two data sets that I have for bone marrow, they have the same number of genes, but for the second data set, I would have more cells. And for the rest of my presentation, I will be mainly showing the results for the first data set of bone marrow. So first I did is QC and data filtering. So I plot the violin plot for the first data set, and I see that for the number of features, the maximum is actually 2500, and so I did a filtering by only including the cells that are having from 500 to 2500 features, and the maximum for mitochondrial percentage is below 5%. So I also set my filter just to keep like cells below 5%. And after the data filtering, I also visualize the highly variable features. Before doing that, I convert the ensemble ID to the HGNC gene symbol, and those ones that are labeled are the top 10 highly variable features in the first data set. And after this, I also did scale the data. And the reason that this is, I think, is really important is that single cell data set contains various sources of unwanted variations. Some might come from technical noise like batch effects. Some others might come from biological variabilities like difference in the cell state. So by normalizing the data would help us to mitigate these effects, which would benefit our downstream analysis. And after this, I did the PCA, and in order to determine the number of principal components to include, I used the elbow plot, and I choose the PCs to be 15 because I think after 15, including more PCs does not really increase does not really explain more variability in the data set. So I set my PCs to be 15. And after the PCA, I did the clustering by setting the PCs equal to 15. I tried different values for resolution. And I tried from range 0.1 to 1, and I see like by increasing the resolution value, we do get more clusters. But for example, when we have resolution equals to 1, we have 13 clusters, but by having this number of clusters, we do see some overlaps between different clusters. So for my downstream analysis, I decided to use resolution equals to 0.3, which gives me 7 clusters. And after that, I did the visualization of the clusterings. I tried different metrics, the cosine and the Euclidean one, and based on these two plots, I think using the cosine as my metric gives a more distinct separation between different clusters. So I decided to use cosine as my metric in later analysis. And I also did t-SNE just to like compare the different nonlinear dimensionality results. And I tried different perplexity, and for this one, I think gives me a pretty good result for having perplexity equals to 50. But comparing to the result given by UMAP, I still think UMAPs gives like a better visualization of different clusters. And with that, I also I did the find all markers, and here is a example for cluster 0. After I did the find all markers, I filtered to only include unique markers. So like the marker genes only for unique to different clusters. I also only include the markers that are significant by filtering out having only p-values less than 0.05. And here
I have to wrap up, Xingyue.
Okay, and here are the example for cluster 0. I look at these for markers, and I used them to determine the cell type. And here's my final result by having 7 clusters, and I did the same thing as I did for cluster 0 for all of the remaining 6 clusters, and I got these 7 cell type annotations.
Thank you. All right, thank you. And let's move on. And next speaker is Katerina. Please share your screen.
Hello, so I'll be sharing my results for the bone marrow and pancreas datasets today. Oops. Okay, so first I'm just going to share my pipeline and how I approached this problem. I've never done dealt with gene expression data, so this was actually really interesting, and I really got into the project the last few days, so it ended up being a lot of fun. So first I loaded the data, of course, and then I did the QC, normalization, feature selection, PCA, and clustering. This was definitely the most time-consuming part, as I really had to play around with the parameters and get a good sense of how each parameter and how I might change them to change the clustering results. Then I did initial data visualization. And so basically I packaged all of the cluster for each cluster result into its own separate CSV file. And then I looked at the top expressed genes in each cluster. And I would then do my own research, figure out what gene names they corresponded to. So for example, this was one of my clusters in data set. In one of the bone marrow data sets, and pretty quickly sometimes it would be obvious what kind of cell type that cluster might correspond to, like within 15 genes for this one. It was pretty obvious this was going to be a natural killer cell, and so I'd name it that. Sometimes I had to do more research or look at more cells, and then I went back and re-annotated my UMAPs and my t-SNE plots. And so the parameters I came up with were 3,000 for the features, usually between 2,000 to 5,000 I found most reasonable, about 60 for the NPCs, dimensions 1 to 25, and so on. Interestingly, the parameters ended up being pretty similar between the two data sets, which made things a little bit easier. I think the only difference was I increased the resolution slightly to tighten up the clusters on the second data set. And then here's what I came up with. So I ended up having about, I think, 15 clusters for one and 18 clusters, or 15 clusters for the first data set. And so I found most cells. I didn't know exactly, like I was kind of flying in the dark because I didn't know how many cell types I might have. So I just went with like what cluster results were the tightest and made the most sense to me. I liked the UMAP plots a little bit better because they seem to be just tighter and more compact than the t-SNE plots. So I kind of went off the UMAP plots a little bit more. And so here's my bone marrow data set 2. Moving on to the pancreas data set. I followed a pretty similar pipeline. I loaded, cleaned the data, normalized it, looked at the data. I had fewer clusters for this one, so it was a little bit easier. I also identified the gene markers for alpha and PP cells, and I checked whether they were even in my data set because that would give me a good sense of like what to look for in the violin plots. And then of course I did a UMAP and t-SNE. So yeah, so first I overlaid the alpha cell and PP cell markers on the UMAP and t-SNE plots, and I was like, okay, they exist somewhere in this data set. I don't know if they'll be in a cluster, but I know they're there. So that gave me a pretty good idea that like a pretty high level of confidence that I might find at least one of them, especially the PP cells. And then I also saw that again in the violin plots. And then lastly, I again did the parameters. And the parameters ended up being pretty similar, except I ended up going with a smaller resolution and dimension size as compared to the bone marrow data sets. I also went with the Euclidean metric for all of them. I just found that the clusters were tighter under Euclidean versus cosine, which was interesting. And then lastly, I annotated all the clusters, but the important thing to note is I did end up finding PP cells, or this lone cluster here at the bottom, but I did not end up finding alpha cells.
Thank you. Thank you, Katerina. Yeah, that's very detailed analysis. And the next speaker, Jiahang, please share your screen.
Conceptual story, can you let the next person go first? I think my first one is block of facilitator. Okay, I just updated the system.
Sorry, I cannot hear you very well. Are you saying that you go next? You'll go later?
Yeah, I think that pretty much the command is like essential to block the function not into like
Okay, now sorry, I still couldn't understand you fully, but I think we can let the next speaker go first. Juliana, please share your screen.
Okay. One second. Okay, can you see everything okay?
Yes.
Okay. So today I'll be talking about my final project presentation for this class. And so I specifically wanted to present about the pancreas data set just because of interests of time. So this data set was the healthy donor pancreas tissue data set. And then I was specifically looking for these pancreatic polypeptide or PP cells as well as alpha cells. And so I used the cell marker 2.0 database that I think a lot of other people used as well. In addition to this paper that I found that talked about identifying marker genes for specific cell populations in human pancreatic cells. So I narrowed down on kind of a couple genes. So for the PP cells, I was looking for PPY expression. And alpha cells, I was looking for GCG, GC, and TTR gene expression. But then I also knew that there are also other common cells in the pancreas, like acinar, beta, delta, endothelial, and stellate. So I also kept that in mind that those might also be present in this data set. So just to kind of show what I tested out. So here's like an overview of all of the parameters that I tested. So I just played around with a couple and then assessed how clean the clustering was. But for some parameters, I did it based on things like the elbow plot, which helped show like which number of PCs were most optimal. So for example, here I found that around 20 PCs gave me like the best number in terms of like the variance explained. But then in other parameters, it was more, I guess, subjective. It was based on like my feeling of how happy I was with the clustering. But then overall, after testing all these like different combinations, this was the workflow that I decided to go with. So for the QC, I went with the minimum cells as 3. I narrowed down for cells that had at least 200 end feature RNA, but less than 2,500 to kind of remove any possible doublet cells. And then I went with a scale factor of 10,000. I chose 4,000 end features, 50 MPCs, 20 dimensions. I tested a variety of resolutions for my clusters just to see like how I just was curious about how the clustering would change with the resolution. I found that the algorithm didn't really change the clustering results, which was interesting. And also I found that in my case, I actually preferred the cosine clustering. I found that it led to like more separation between the clusters versus Euclidean. So here are the results for the final clustering. On the left is the UMAP visualization, and on the right is the TISNE. I think in general, I preferred the UMAP as it led to like very clear separation of like 0, 1, and 2 clusters, whereas the TISNE, it seemed to be a bit more bunched together. And then from there, I just looked for the marker gene expression of the genes that I mentioned before. So interestingly, I found that the alpha and PP marker gene expression seemed to be concentrated in this small cluster, which was cluster 2 right here. And then all the other clusters 0 and 1 showed high expression of this reg1a gene. That's a marker for like an acinar cell type. So I was thinking that perhaps this one cluster featured both cell types. So I decided to go with subclustering analysis. So I just extracted that specific cluster and did the same kind of workflow as in the original data set to see if I could separate out that one cluster. And I think that it was modestly successful. Not really. It didn't separate as cleanly as I would have liked, but I think one thing I noticed was that it seemed that the PPY, like strongly positive cells, were more close to the bottom, and then the ones that the alpha cell, the cells that were positive for the alpha markers were more closely bunched toward the top. So there seemed to be some level of separation. But I also saw there are a lot of cells positive for this INS gene, which is a beta cell marker. So going along with that annotation, I also use violin plots to be more, to further validate my predicted annotations. So this is what I ended up with at the end, where I put these two clusters I denoted as those acinar cell type, and then I had my alpha, some beta, and some PP. And then just as like a sanity check, where I was just curious, I tried predicted annotation using single R. And I feel like for the most part, it lined up very well. I think they actually found that there was more beta than PP. So I might have like switched those analysis. But yeah, I think in the future for refinement, I would definitely look at more than a single marker genes. I would try to look at multiple. But I think for the most part, it worked out relatively okay. So yeah.
Thank you so much. Yeah. Thank you. And our next speaker is Hannah. Please start sharing your screen. Yeah. And you're muted.
Yeah, I should be. Am I all good? Can you guys see my final project?
Yes, we can
Okay,
see it.
perfect. I did a pretty informal preparation, so sorry for not having cute slides ready. But essentially I was just going to show off what I've done for the pipeline so far. For full clarity, I did start off pretty heavily on using like the standard pipeline tutorial, and I haven't tweaked too much farther from that. But here's my imports. These are my QC plots for both the feature and the count RNAs. And this is actually for bone marrow data set 1 and actually bone marrow data set 2. I did all of them. And then this is honestly a little zoomed in. This is another one of the example QC plots they have where you can see the end count RNA as well as the feature RNA, and you can see which ones have like way too much. But this is after filtering. And then here is where I subsetted the stuff. This is subsetting for cells that have too low counts and cells that have too high counts, because the cells that have too low could be dead cells and cells that have too high could be doublets. So that's filtered out. I didn't do the mitochondrial ones simply for the fact that we were given ensemble genes. And I didn't want to convert the ensemble genes. But that generally is something you would want to do as well. I chose to do log CPN normalization on both of these just because it's something commonly done. I use the standard parameters for this one using n features at 2000. That's just what the tutorial gives. And then I actually did the variable features pretty high up because that's where they did it in the tutorial. And so this is the variable features with the labels for data set 1 and for data set 2, like that. Let's see, we're probably gonna want to zoom in a little bit more. And then for this is scaling, which you have to do before doing PCA. Here I ran PCA again with the default parameters here. And these were the plotting results of that. And I also plotted heat maps, but honestly, the heat maps are pretty hard to read given ensemble genes because they all start with ensemble and then they don't get to the point where you can use them. So ideally, if I were to do this again, I would convert the ensemble genes, but I haven't done that yet. And then here are the elbow plots, which I use to determine the like proper embedding dimension. Honestly, there was like, a lot of good answers here, but I ended up picking 15 because it felt, at least from where I had this cropped originally, which was at 20, it looked like the elbow. I think 10 is also a good option, but up to 20, I wanted to be a little conservative because I don't like removing too many PCs personally, just for the sake of making sure I have most of the information. Okay. And then for clustering, notably, I used dimensions 1 through 15, which is what we had seen from the previous plot. But the main thing that I did differently from a lot of my peers is I actually used Leiden clusters instead of Louvain, just because I prefer Leiden. I think I've had more luck with its stability, but that might just be me. And then I also used Tisney instead of UMAP because again, I prefer Tisney to UMAP personally. Yeah, so this is for bone dataset 1, bone dataset 2. And then I haven't really looked through the results for find all marker genes yet because I was trying to manually annotate them and then go back and validate with that. But you can do a heat map for this, also illegible because of the ensemble genes. And finally, I was in the middle of trying to annotate the clusters using given ensemble genes, basically trying to find which clusters they go to. So these were some of the notations I was taking, but that's the part I haven't done yet. So I did pretty much like all the standard pipeline for data sets 1, 2, and also the other data set, the pancreatic data set, but I didn't get to fully labeling everything yet. So that's where I'm at. Yeah.
Thank you.
Thank you.
Tianheng, are you ready to do your presentation or you have to wait longer?
Jessica, can you hear me?
Yes, a little bit noisy.
Okay, yeah, I guess this is the best way. I'm calling through my phone, so but I guess this is the only way out.
Okay.
Let me try to share the screen. Can you see my screen?
Yes, we can see it.
Oh yeah, so I didn't expect it to be a full slide presentation, so I just put it in my document. So what I did is very typical first step is to read data and do PCA. Initially, I also have the quality control in there, but because I suffer from constant oversplit, and I was wondering if I did a too strict criteria on quality control, so I removed it on this code. And then initially, I tried to use a silhouette to find the optimal resolution. But then I also tried the criteria by selecting the resolution to maximize the number of marker for clustering. But the final version I'm currently sticking to is I use a hierarchical clustering with maximal distance-based consensus clustering for determining the clusters of myself. And this is the plot. So I only use the first data set as the illustration here. The procedure that I carry is very similar for the first two data sets. So as this plot shows, the first cluster, the first data set, I determined that 16 cluster might be the best number. And this is my visualization with the UMAP. I like Tisney a little bit better, but specifically just for the first data set, I think UMAP is showing better what problem I encounter. So most of the cases I have like these very small clusters, which I have to determine whether I should merge them or not. So then I proceeded to heat map plotting. Here are all the gene expression of the markers among all the clusters. So there are two ways that I mainly use. The first one is the eyeball if there are high similarity between two clusters like cluster 2 and cluster 6. And also there are two clusters, for example, cluster 11, cluster 14. But consensus clustering result is already not most obvious in terms of over clustering. So I further did a little bit about FDR controls that identified between cell type DE genes for potential merge between clusters. But one phenomenon that I noticed is that sometimes if I have a group of clusters that I want to merge, most of them may relate to the multiplication of the cell type. So for example, if 2 can merge with 8 and 2 can merge with 9, but 8 doesn't merge with 9. So this is a potential thing that I was still trying to figure out if I should count them as the same cell type. Because in our data set, the cell type we're given is very coerced instead of like a progenitor or like very specific stuff. But anyways, so that's for the first two data set. And for the pancreas data set, what I was given is a really large chunk one. I think in the instruction, we were specifically instructed to use Surette to do the clustering and cell type annotation. But I think what I remember and also verified online is that because gamma is a very minor cell type, the Surette clustering doesn't really tell very well about the cell type detection, the minor ones. So I was looking into if I can use some other ones to see which one is more like a PP gamma. So this is my current progress.
Okay, thank you. Thank you, Jahan. All right, our next speaker is Melanie. Are you here, Melanie?
Yeah, hello.
Hello, yes, we can hear you. Could you
Okay,
share your screen?
yes. So my presentation is gonna be very brief.
No problem.
I just started and I spent a lot of the weekend playing around with the parameters. So to give some background, I don't really know R very well. I don't really know Seurat very well, which is maybe why this pattern is in the best shape as of right now. But the first thing that I tried to do was to run the first two steps of the pipeline to output up all the way up to the HPG part, according to the R from the Boolean on R, like the surrounding it. So from there in the HPG for so long. So this is HPG for so long, so I kind of just threw out that data set, but in the future I would use these HPGs to validate my current results. So from there, I just Googled different so clearly I analyzed concrete results. So I just Googled different microgenes that were already known that started from to identify a cluster from there. Um so these are the baseline parameters that I use to start with. K-pyramid 500, and later on in the presentation I'll show like how I changed K-pyram to a bigger value to see to validate whether my clusters were correct. So just to foreshadow, I changed the resolution, I changed sorry. I changed the variable features in the first step in I used SC cancel. So I changed the variable features. I also changed K-param, UMAP numbers, and resolution. Right? So this is just a plot I made literally an hour ago just to let myself know that I need to do QC later because I kind of forgot about that. When I ran the pipeline based on Group 10 presentation on Wednesday. But the first thing I actually did was to try to see if I change the number of variable features in SC transform, can I use the markers that I googled to sort of find out different like the best number of variable groups. And what I ended up going with SC transform variable features is 3000 because I thought the graphs looked the most well rounded. So I just went with that. And then I've said, okay, so I like started with 3 clusters so far. That's not really like what I want. There's only supposed to be 2 clusters because we're supposed to identify 2 cell types. So maybe like if I play around with the K parameter because in the graph on the left over here is the original 3 clusters. No, I was hoping that because the marker genes on the left are specifically for like PP cells, I think. That would mean that 0, 1 would sort of 2 different clusters for the alpha cells. So I tried to like kind of decrease the parameter to see if I could force those clusters together, and maybe that would justify my clustering result. Again, not very good, but I try to use the markers to like cluster, right? And then from there, when I felt satisfied with what I currently had, I grabbed the UMAP of it. And I grabbed so on the left here, on the far left, you could tell it's 3 clusters. That's when I had a K parameter of 1000 in the original was 3, and then I played around with like the like K, so the bigger the K, I feel like the more defined the clusters are, because you can see like over here, like yeah, I guess the more I guess now that I think about it, it's kind of the same graph because it's just like rotated a little bit, but yeah, I kind of and so I did I did play around with the resolution. And I found that for bigger and smaller resolutions, I got very like the bigger the resolution, the more clusters I got. So I just kind of like threw that RAM. And the smaller resolution I had, the less clusters I got. So I just stick with the 0.8 parameter. And for future work, first of all, I want to try qualities and control based on the graphs that I put in the earlier slide, and I'm gonna print out the HPG graphs just to see like do my current results match with the HPG, and if they don't, what can I do about it? And I could also plot a heat map of all the PCs and use find referencing by cluster to really verify my results.
Thank you, Melanie. One thing I would like to suggest, which I don't see in your results, is PCA. Did you do PCA?
Yeah, I did. Oh wait, actually, I may have. I think so. I may have forgotten about the part.
Yeah, maybe that's the part because you did highly variable genes, but then you have to go through PCA and then find neighbors. Maybe that's the thing.
Oh, yeah. Thank you.
Okay, thank you so much, Melanie. I'll move on to our next speaker. Koa, is that the right way to pronounce your first name?
Yeah, it's Qual, but it's fine. So let me just share my screen really quickly.
Okay.
Can you hear me? Can you see my screen?
Yes, I can see your screen.
Okay, great. So again, my name is Qual Vu, and this is my final presentation. So starting off, quick pipeline overview, reading in the RDS. So I ran the RDS single cell RNA sequencing object using read RDS. I converted RDS objects to Seurat objects using create Seurat object and filter out genes that appear in less than 3 cells and filter out cells with less than 200 features. So my presentation will be code on the right, so just to help people follow along. Next up, ensemble to HGNC. I used Biomart to retrieve HGNC genes for each ensemble ID present in the RDS object. Again, I noticed that some ensemble IDs do not have a corresponding HGNC gene ID. I found ensemble IDs of mitochondrial genes to calculate quality control metrics. I calculated, next up is performing quality control. I calculated quality control metrics with percent featured set. And the features were the ensemble ID list of mitochondrial genes. I then visualized the quality control metrics with a violin plot and kept cells that met my quality control standards for bone marrow 1 and 2. I kept cells within 200 and 3,000 N feature RNAs and less than 8% mitochondrials. And for the pancreas dataset, I decided to do 200 between 200 and 4,000 end feature RNAs and less than 8% mitochondrial. These were some of my quality control violin plots. To the left and the bottom, at the top right, is bone marrow 1 and bone marrow 2. As you can see, for me, it was largely 8% mitochondrial here and a little less than 8% for both data sets. That seemed to keep a good amount of cells while we're kind of removing these outliers here. I don't know if you can see my laser pointer or my cursor, but I hope that it's at least, you can at least follow along to my voice. But yeah, and then 200 and between 200 and 3,000 end feature RNAs seem to be a good point to, again, keep most of the cells while removing the outliers. For pancreas, the only thing that really changed was end feature RNAs varied a lot more from 0 to 6,000. So I decided to move up the threshold to around 4,000 end feature RNAs. Next up is normalization and feature selection. I use SC transform for normalization and feature regression, or for feature selection, and I regressed out percent mitochondrial. So I ended up not using scale data and fine variable features. Next up, PCA, UMAP, and clustering. I set the C to 1 for reproducibility. I calculated PCA with run PCA, calculated UMAP with run UMAP, and used the first 30 PCA dimensions. I calculated neighbors with find neighbors and identified clusters with find clusters using the Latent algorithm, which is algorithm equals 4. I plotted the dimensions using dimplot. And for marker genes, I found cluster-specific genes with find all markers. And I used the marker genes of applicable cell types from the Surratt's tutorial BigNet, which has a good amount of the cell types that we're looking for, like CD4 T cells, CD8 T cells, and B cells, and monocytes, natural killer cells, and dendritic cells. The rest of the marker genes I kind of derived from literature here and there. And next up, finally, I annotated the cells. This is for bone marrow 1. I ended up with around 13 latent clusters and 9 unique cell types, being CD4 T cells, CD8 T cells, CD16 natural killer cells, plasmacytoid, dendritic cells, naive B cells, myeloid progenitors, classic monocyte cell, dendritic cells, and non-classic monocyte cells. These are some of the clusters and their annotation labels on some of the marker genes I use, but I feel like I will go more, I'll write all these, and due to time constraints, I will be going over all of them, but they'll be in the final report. For bone marrow 2, I had 15 latent clusters and 10 unique cell types, those being plasmacytoid dendritic cells, dendritic cell, classic monocyte cell, myeloid progenitor, plasma cells, precursor B cell, naive B cell, CD16 natural killer cell, CD8 T cell, and CD4 T cells. Again, these are just some of the clusters and their mappings. For pancreas, it's currently a work in progress. I have 8 latent clusters. Tentatively, I decided for the pancreatic polypeptide cells, the marker gene I'll be using is PPY, which is plotted to the right of the latent, the latent UMAP. And for alpha cell marker genes, tentatively, I decided to use ARX, MAFB, and GCG, which are listed below, respectively, in the bottom UMAP. And yeah, that's it. That's all I had to demonstrate.
Thank you. All right, our next speaker is Emma. Please share your screen.
Sure.
I'm sorry, let me
You're good.
unshare. Yeah.
Okay, so for my first part, I had bone marrow data sets. I started out with just using purely default settings, and then I think I'm going to go back and rework it with more fine-tuned parameters. But with the defaults, I had 18 clusters for the dataset 1, and then 12 clusters for dataset 2. I found the top 10 highly differentiated markers for each cluster. And then using these top 10 genes, I put these into this ACT database, which gave me this map for what the dataset predicts the cell to be. And overall, I ended up having these assignments. So the T cells kind of cluster together with the natural killer cells, the monocytes cluster together, and then the B cells cluster together. For part 2, I did a little bit different approach, but I still used the default parameters, which gave me 12 clusters again. Instead of using their search tool, I pulled the cell markers from ACT for the PP cells and the A cells, and then created violin plots for both of them. So these are the marker cells for the A, marker genes for the A cells. I came to the conclusion that 7 and 8 were the most likely to be A cells. There's few genes where they were the only cells with these genes being expressed. And then for the PP cells, I didn't find any, I didn't come to a conclusion that there were any PP cells based on this evidence, but I might go back and look more and refine the settings a little bit and then rerun this analysis. So this is where I think the alpha cells are. And that's mine.
Thank you. And the next speaker is Andrew.
Okay, hello, can you see my screen? Okay, so yeah, I'm mainly going to present on the first bone marrow dataset in the interest of time. So the first thing I did with this dataset was I used gprofiler 2 to convert the ensemble IDs to gene symbols, just for like convenience sake in my downstream analyses. And then for the preprocessing and quality control step, I used mostly default parameters. I had min cells set as 3, min features set as 200. For normalization, I had a scale factor of 10,000. For HVGs, I kept the top 2,000 highly variable genes. And for run PCA, I mainly plotted the first or the 30 PCs because when I looked at the 51, the 30 through 50 look kind of redundant to me. And here's the elbow plot for PCA. I did choose a lower number of PCs to maintain for my downstream analyses than most of my peers because that the red dashed line is where I personally thought the biggest bend in the plot occurred. But I could definitely see an argument for like keeping say 11 or 12 PCs. And then for clustering, so on the left here is my initial clustering. I first did my parameters were prune SNN equals 1/10, K parameters equals 15, and a resolution of 0.8. And I was initially working with this clustering here, but as I guess did my annotations, I found out that a lot of these clusters had overlaps in marker gene expression with each other. So I decided to go back to my clustering and then tune the parameters a bit more, and I ended up with the parameters of prune SNN equals 1/15, K parameters equals 20, and a resolution of 0.5. And this is the final clustering right here on the right. And so when I was tuning these clusters, I guess the main thing that drove me to change my parameters was when I was making these feature plots of known marker genes for cell types that I thought would be in my data set. This here is an example of GZMK, which is a gene marker for CD8T and MAIT cells. And you can see that the expression of GZMK is correlated, the decent correlation with cluster 4 in my final clustering. So that gave me a little bit of confidence in my final clustering. And similarly, looking at another marker gene, CD14, this is the marker for classical monocytes. You can see that the expression of CD14 has a pretty strong correlation with cluster 0 in my final clusters. And so moving on to my annotation, I guess pipeline, the first thing I did was I used find all markers, and then I sliced the top 10 marker genes for each cluster. I think I sorted by p-value. And then I did violin plots with these markers to check differentiation and expression to make sure these were, I guess, uniquely expressed within each cluster. And then after that, I cross-referenced these markers with literature and external sources like research papers, the Azimuth bone marrow reference, Human Protein Atlas, and this app called the Absolute Immune Cycle Deconvolution, which kind of just you input a gene and then it gives you a potential cell type, immune cell types that it could be found in. So I use this tool mostly for immune cell types. And so here's kind of an example of an annotation that I did. If you remember from a few slides ago, CD14 is a marker gene for classical monocytes, and it was strongly correlated with cluster 0 in my dataset. So when I did violin plots, the same pattern appeared in these violin plots. So I had a pretty strong evidence that cluster 0 corresponded to classical monocytes. But to make sure, I also plotted another marker gene, APOBEC3A, which is also actually a marker gene for non-classical monocytes. So the expression of this gene in cluster 10 gave me some insight as to what cluster 10 could be. And then to kind of confirm my suspicions about cluster 10, I plotted a violin plot of LYPD2, which is a marker gene for non-classical monocytes. And then that kind of confirmed my suspicions that cluster 10 is a non-classical monocyte because it is preferentially expressed in cluster 10. So just kind of using this pipeline, I did this with every cluster I had. I believe I had 12 clusters, and these are the final annotations for my first bone marrow data set. I'm pretty happy with them. Just the main, I guess, thing that I do want to kind of go back and check on is the top right cluster here. That's currently classified as a CD8 T cell. I think there could be 2 clusters in there, CD8 T and MAI T cells, but I will probably need to go back and work on the clustering to make sure about that one. Yeah, that's it. Thank you.
Thank you, Andrew. Yeah, it's very careful analysis. And next speaker, Shirley.
Okay. So I think I'm mostly going to talk about the bone marrow ones because my pancreas data is not looking good right now. But for the bone marrow ones, I followed pretty much the very standard Seurat pipeline. I read it in, created the object, and then I created the violin plots. And from here, I decided to get rid of the values where the end feature were above 3750. And for end count, the ones that were above 20,000. And this kind of reinforced that like these values needed to go up here. So I got rid of that. I normalized the data. For a lot of this, I kept default parameters because I think I was trying to make some sense of what was going on step by step. But at this point, in hearing from everyone else, I'm going to go back and kind of mess around with it. But I normalized it using the default. I decided to identify the variable features, took the top 10, which we can see here, scaled the data, and then I performed PCA and used an elbow plot to see how many dimensions to use. And I think I'd settled on 10 because I know it drops off here, but I figured like this is where most of the curve was. So then I clustered the cells, again using 10 dimensions. And these are my clusters. They don't look the best. I have to go back definitely and mess around with them some more, but I feel like for the most part, it was able to do a decent job. And then I followed, once again, following the pipeline just to find the differentially expressed features. And I think I ended up with, yeah, 12 clusters. And then what I did is I used the BioMart package to convert the top 10 for each cluster into these. And then I went through and I researched on myself to try to figure out what each cluster was. And I did that using, I forgot the name of it. It was some database online. But I went through, searched on my own, and then assigned the cell types to each cluster. And some of these make sense. Like I don't have a bio background, but to me, it makes sense that all these T cells are going to stick together, like these B cells would be together. What doesn't really make sense to me is both of these were defined as dendritic, but this one was this type of dendritic, and this one was like the normal kind. So I'm not sure if that's supposed to happen, if that's normal to see, but that's something I need to look into a little bit more. And then I did the same thing with dataset 2. And I kept the same cuts based off the violin plot just because it looked pretty similar. I'm going to skim through this because a lot of it is the same. I think the main difference is for my dimensionality. I decided on 15 here because it's kind of flat, but then this little drop here kind of got me thinking. So I decided to do 15. And this one ended up with 13 clusters, I believe. Yes. And again, it's not the best. I have to go back, but these were the clusters. And then same situation. I looked at the top 10 for each cluster and I went through by myself and tried to figure out what each marker meant. And then I just assigned them. And again, the T cells clustered together, the B cells were together. And then just this doesn't quite make sense to me. It's the dendritic ones, but that's where I'm at. Oh, and there was one I wasn't able to identify. This one here, I'm very confused on, but
yeah, I have to go back and look more into it, but this is where I'm at right now.
All right, thank you. Thank you for the presentation. And next speaker is Delina. Could you start sharing your screen?
Yes. Okay. Can you guys see my screen?
Yes.
Okay. Okay, so hi everyone, my name is Delina, and I'll be presenting my analysis for the Seurat pipeline. And then the goal was to identify and annotate different cell types using, in the dataset using the Seurat pipeline. So just for an overview to start, the dataset consists of single-cell RNA-seq data from human bone marrow tissue, and I focused on the dataset 1. The dataset includes a variety of cell types, such as monocytes, T cells, B cells, and more. However, the actual number and types of cells included in the dataset remained unknown, so my job was to classify each cluster from the 21 given cell types. So to begin, I started by pre-processing the data using SC transform for normalization, which also handles scaling and highly variable gene selection. And then next, I performed dimensionality reduction using PCA and clustered the cells using the find neighbors and find clusters functions in Seurat. I visualized the clusters using UMAP, which revealed 19 distinct groups of cells, as we can see on the right-hand side. And then after this, to annotate these clusters, I identified the top 4 marker genes for each cluster using the find all markers function, and then manually annotated the cell types based on the known marker genes from literature. And then just to go into like detail a little bit more, so the top 4 marker genes for each cluster I used were selected based on their fold or log fold change values using the find all markers function in Seurat. So initially all marker genes were identified, and then the gene identifiers, known as like the ensemble IDs, were mapped to their gene symbols using the biomart package to ensure the unique mappings by removing some of the duplicates. And then finally, the top 4 marker genes for each cluster were determined by grouping the marker list by cluster and then selecting the highest log fold change genes. So in this case, it's the highly expressed and their characteristic markers for their respective clusters. So after identifying the top 4 marker genes for each cluster, I validated the clustering results using violin plots and feature plots. So just for an example, let's say cluster 5. So the top 4 marker genes are listed here. And then these are known markers for neutrophils. And then first I used violin plots to confirm that these genes were differentially expressed in cluster 5 compared to the other clusters. As you can see here, it's highly expressed in cluster 5 compared to the others. So the next I visualized the expression of these markers using feature plots, which allowed me to confirm that the genes were specifically expressed in cluster 5, and also ensured that the clustering results from what we obtained from the previous step was accurate. So based on the expression patterns of these marker genes, I annotated cluster 5 as neutrophils, and then this process I repeated for all 19 clusters to ensure accurate cell type annotations. So after annotating the clusters, I identified cell types such as the regulatory T cells, monocytes, CD8 T cells, and more. So for example, cluster 0 here, I annotated as regulatory T cells based on the expression of marker genes like FOXP3, and then cluster 1, I annotated as a monocyte based on the expression of marker genes like CCL2. So on the right here is the final dim plot showing the annotated clusters, which each color representing a different cell type. I just wanted to go over some challenges. So one of the ones that I faced was ambiguity in marker gene expressions of some clusters, especially for closely related cell types like different subtypes of T cells. And another was determining whether a cluster was overclustered or not. So to address this, I tried to play around with parameters like resolution and find clusters and k-dot per m and find neighbors to ensure that the clusters had biological meaning. But despite these challenges, I was able to annotate all of the 19 clusters I identified. But I still think there's room for improvement in my code. And then on to conclusions. So I was able to annotate all 19, but I believe that I could, these are just preliminary results I generated only for one data set, so I'll have to apply this to the other full data sets for the project. But I do want to explore possible overclustering as shown in the red box here from the original Seurat clusters generated, and then maybe try to validate some of the marker genes using additional data sets and try to refine my clustering parameters to improve my confidence in these annotations. Thank you.
Okay, our next speaker is Ziheng. Please, Ziheng, please start your screen.
Yeah, let me first share my screen. Okay, see my screen. Yeah. Hi everyone, and now I will introduce my analysis of this single cell data for this bone marrow data set. And I think my whole pipeline will be very similar to other students' work before. And our goal is to just identify different cell types for the whole data set. And this bone marrow is just use the UMI count matrices, and there are possible like 21 cell types. And then I will use the dataset one as the example. And so this 21 cell types. And so my whole process starts by like loading the dataset and creating this object. And I normalize the data, find some highly variable genes, and also perform the PCA and some neighbors. And finally, I use a UMAP to visualize the cluster. Many clusters I used is just the default one. And I think the UMAP also can help me to group cells and assign some different types. And so for the cluster, there are total 11 clusters, and the process is the next slide. And I mainly use the UMAP plot to see whether the tuning parameter is better. And in the UMAP plot, I observe total 11 distinct clusters from 0 to 10. And some clusters like 0 and 1, they are large and well separated, which means they may have some distinct cell types, but also there are some smaller ones like 6, 8, and 9. So that means they may have some very rare population, and also for some like closely clustered one like 2 and 5, they may indicate the biologically related cell types, but I will I will need to check it further. And next, so for the single cell type, the annotation, I use the find or marker genes function, and I also use and then after I use this function, which it can identify the genes that are very significantly expressed in each cluster, and then I organize the results by grouping them and selecting the top 100 marker genes based on their adjusted p-values. And then I use this ensemble database and map the gene IDs to their gene names for the next steps. And then after classing the cells and analyzing marker genes, I just assign these labels to each cluster. And this table summarizes this identify the cell types for each cluster. And by comparing the expression of the marker gene with I found this marker genes for each cell type from some online reference. And then I just determine this from last slide, after selecting the 100 marker genes for each cluster, I just compare each marker genes of each cluster with the marker genes of this 21 cell types. And then I just determine which cell type has the most matches with this given cell type. For example, if we select like 10 marker genes for each cell type, then I just compare like maybe this one, it have like the number of match. And then so that cell type is assigned to this cluster. Yeah. Thank you.
Thank you, Ziheng. Okay, our next presenter is Yuxing. Are you here? Please start.
Hi, can you hear me?
Yes.
Let me share my screen. Can you see my screen?
Yes, please start.
Yep. Hi, everyone. I'm going to present my final project. And I just doing the analysis follow a classic CREP pipeline, and I start with a quality control. I draw this count versus feature map, and you can see in the first map, there are some outliers. So I just filter out all the data with count number larger than 22,000. And for the bone marrow 2 the dataset 2, I didn't do any change. Then I do a very classic normalization, data scaling, and find feature step and run the PCA. And after running the PCA, I draw this elbow plot. And for the dataset 1, I choose the first 14 PC, and for the second one, the second dataset, I choose n equals to 17. And I perform the UMAP to draw this UMAP plot. And here is a result. For dataset 1, there are 12 clusters. And for dataset 2, there are 14 clusters. Then I'm mapping each cluster with the marker I find online for each cell type. I will show you a couple here. First is CD14, which is a classic marker for classical monocyte cells. And you can see here for dataset 1, cluster 2 here are classified as the classical monocyte cell, and in dataset 2, the cluster 0 are classified to this type of cell. And for this one, it's the FCER2, which is a marker for naive B cell, and in dataset 1, cluster 4, and in dataset 2, cluster 8 are classified into this type of cells. And for CD1C, which is a marker for dendritic cells, and cluster 9 and cluster 12 in dataset 1 and 2 are classified to this type of cell. And here is the overall plot of the results that I mapped for dataset 1 and dataset 2. And these are the results for the part 1. And for part 2, I marked the PP cell with the gene marker PPY here. And you can see here, I'm just doing the similar process with running the PCA and then the UMAP. And for most of the cell, they are just irrelevant cells. And for these two types, which are the PP cell and alpha cell, I classified. And for the PP cell, you can see there is a very high level of PPY expression. And for alpha cell, I choose IRX2 as the marker, and they got a very high expression here in alpha cell. I classified. And that's all for my presentation.
Thank you very much. Yeah, the results seem quite good. And the last speaker is Tom, and our very last speaker of the whole session. Please start sharing your slides.
Oh yeah, so can you can you see my screen? Okay, yeah. So today I will present my project. It will be includes two parts. First is the data processing, and the second part I will show the result. So first, I do the quality control by filter out low quality cells and genes based on some specific feature counts and cell counts for all those three data sets. And then I normalize the data by using SC transform method and also the normalized data method. I use SC transform for the first one and the normalized data function for the left two data set. And after that, I did the dimensional reduction with PCA. So I also use the elbow plots to help me find which is the optimized number of PCA. So like for example, for this one, I will choose 20 as my PCA number. So here is the parameter I chose for the bone marrow one dataset. Yeah, I did try some different parameters like different resolution, but I think those are the optimal one I choose in the end. Yeah, I tend to choose a relative smaller resolution because I want to have more compact and small cluster. So here for the bone marrow one, we will have total 10 clusters, and for each of them, I choose the first 5 top markers to help me like identify what kind of cells it is. So here's the result. And also I did the similar thing for the dataset too. And here's the parameter I chose for the second dataset. So this one, it gives me 11 clusters, and here's the results. Because I'm not very familiar with the bio method, so at least those clusters looks good to me. Yeah, and here this is the parameter I choose for the third dataset. I try to figure out like what kind of did it contain the alpha and PP cells, but I didn't have a very clear cluster here. Yeah, and I'm not sure whether this is correct or not. I just marks the cells which contains those gene markers for PP and alpha cells. And I also did like other clusters for the third data set because I can't get the PP and alpha, and here's another result. Yeah, that's all I have.
Okay, thank you, Tom. And thank you all for the presentations. I hope that you find some interesting results from each other's presentation, and maybe you can use that to help you refine your analysis. And I would say that for single cell data analysis, some knowledge is necessary, right? You need the knowledge to help you guide your parameter choices. There are so many steps, so many parameters you have seen from the analysis. So I would say in particular, single cell data analysis is not yet automatic because every data set is different. And that's why we give you 3 data sets and different knowledge, right? Some of the knowledge you can gain from literature or from online search, but we want to see your honest analysis in your results. So hopefully you find the presentations from other people useful, and you still have some time to refine your results. So that's why we give you some time between the presentations and the report, the due date of the report. So in the meantime, just take it as a practice, as if this is something you're doing research on, and see what you think based on your subjective judgment. That's totally reasonable. What do you think is the most reasonable result? And we look forward to receiving your reports and your results. So thank you all for being with me for this quarter, and I hope that you find the class useful and learn something useful for your future work. Okay, thank you everyone for staying till the end.
Thank
Bye.
you.
Bye. Bye everyone.
Thank you. Bye.
Bye.